So taking the time, and that was only like 5 minutes, taking the time to identify the key vocabulary and see if you can piece them together by writing a summary is key. I think I even mentioned this back in lab when we were talking about studying and the kind of the big picture and one of the best ways to study would be like imagine taking all of your note cards with all of the vocabulary terms on them and shuffling them up and dropping them on the floor and picking up one thing and picking up something else and seeing if you can link the words together. That's how you really know that you're learning, right? Can you make the connection between acetylcholine and a T-tubule, right? Can you make the connection between calcium and an axon terminal? Can you make the connection between a sarcolemma and an action potential, right? Can you link seemingly unrelated vocabulary words together? If you can't, if this looks like a foreign language to you, then you know you're not putting in the time to this stuff. It's super important that you're not neglecting the physiology piece because you're studying for the bone piece. I get it. We all have lives. We're all super busy outside of class. I graded your quizzes while you were taking your quiz today because I forgot. Like, life happens. I totally get it. But this is your grade, and I want you to be aware of the fact that exam 2 is typically the lowest of all the exams for A&P 1 students because of the bone practical butting up right next to it. So there's only 2 chapters on the exam, but there's a lot of physiology. So look at it. An action potential travels down a neuron, arrives at the axon terminal. You should be picturing this in your head, right? You should be picturing this axon terminal, right? When in doubt, draw it out. If looking at all of these words kind of blows your mind, take a step back and draw it out. I know a lot of you are visual, right? Then we imagine this muscle cell, right? Action potential comes down, that excitement, right? This traveling charge. That's going to trigger the influx of calcium, right? Calcium goes in. It's going to kick out all of our little vesicles that have acetylcholine. Now we have acetylcholine floating around in our synapse. That acetylcholine is going to bind to receptors on our motor end plate. The motor end plate is just a special section of our muscle fiber where the receptors are. There's little receptors right here that acetylcholine binds. When the acetylcholine binds to these receptors, sodium channels open. And then sodium can rush in. Right? Sodium rushing in is that depolarization. Right? Remember this? Resting membrane potential, -85, sodium channels open. Whoop, right? That's that excitement that's happening. We go up to +30. Right? Sodium rushes in. As sodium now is filling this area, it's going to spread. This charge is going to spread, and it's going to open more sodium channels and more sodium channels. And guess what? There's little T-tubules all along our membrane, and that action potential is going to spread down there. And when that action potential spreads down by the T-tubules, which are right next to our sarcoplasmic reticulum, tell our sarcoplasmic reticulum to release calcium. That's the whole chain of events.
Is that one like what gated kind of channel is?
So there's voltage, off of the motor end plate, there's voltage gated channels. So it's kind of that domino effect. You're getting excited, so I'm going to get excited, and I'm going to get excited, and further down. And then that action potential just triggers. So
So it's voltage gated? Or?
Yeah. So it's going to trigger the release of calcium from there. Yeah. So let's go through some multiple choice questions. Let's review. The end plate potential is generated by, well, that one, it's already highlighted for us, sodium, right? Influx of sodium into the motor end plate. I don't know why that one was. Okay, acetylcholine is released from the synaptic terminus in response to, what's the best answer here?
An action potential arriving.
The action potential arriving, right? A better answer would be calcium coming in, right? Calcium coming in, kicking out the acetylcholine, but that's not an answer choice. The term synaptic cleft, what's that?
The gap between the neuron and muscle.
Yeah, it's the gap, right? the space, the gap or the space. That's why they're not actually touching here. Sometimes it's just called a synapse. Sometimes it's called a synaptic cleft. Either way, it's referring to that gap. Sodium channels of the motor end plate are right here. Yeah, they're ligand gated. Remember, the word ligand is referring to like a chemical, right? They will open in response to a chemical binding to them. So they're ligand gated channels. The end plate potential. The end plate potential. Right? The end plate potential is that local depolarization. It's just a small change. Right? A small change here triggers the voltage gated channels to open downwind. Right? Small local change, local depolarization. Right? So we're getting into this idea of excitation, contraction, coupling. Right? We talked about did I do this already? No, okay. I couldn't quite remember exactly where I left off. So we did the excitation. That's this. So we are going to couple it or pair it.
Yeah, we got that. Yeah, we did.
We did? Okay, I thought so
We did
too.
all the way up to calcium.
Up to 3,
ions.
yeah.
Boom,
Action
so we did
potential.
this. Oh, we didn't do these questions. I
Yeah.
knew there were some questions that we didn't. Okay, let's go through these real quick. The channels that open the sarcolemma surrounding the motor end plate and generate an action potential.
Voltage gated.
Right. So they surround that motor end plate. Those are our, went the wrong way. And now it's freaking out. There we go. I had a huge trouble with this board today. Yeah, they're voltage gated. So we've ligand gated here, voltage gated downwind, right? The term propagate when referring to an action potential. What is that referring to? Propagate.
Spread.
Right, spreading, right, traveling. But I thought those questions seemed familiar. In order to trigger a muscle contraction, an action potential must reach triads. Remember what a triad is. We didn't mention it. I didn't have it on my list. The triad is this magical area of the T-tubule and the terminal cisterna, right? It's where the T-tubule and terminal cisterna meet up. It's called a triad. Good. A triad consists of, right? It consists of 2 terminal cisterna and a T-tubule. Boom. All right. Blank is released from the SR in response to an action potential. Yeah, calcium. All right, so that was all excitation. We got our cell excited. Remember I said there's lots of pieces of this puzzle, right? We talked about the overall anatomy of a muscle fiber. We talked about what action potentials are and this idea of a sodium and potassium balance to keep the charge distribution. We understand this process of excitation. Now we're going to couple it or pair it with contraction, right? Actually getting our cell to contract. So where all the magic happens. Calcium is key. Calcium being released from our terminal cisterna is what does it all. So basically, once calcium is released, your muscle fibers are going to contract. What calcium does is it binds to something called troponin. So let me draw, our myosin. Remember myosin are thick filaments, right, which have these like little globular heads. Kind of look like sideways golf clubs, right? And then in between them is actin. So actin is our thin filament, right? But it's made of some globular things, right? So it's not just a line, right? There's actual globby pieces on there. And there's something called an active site. So in other words, there's a specific spot that our myosin needs to touch or needs to meet up with in order for this contraction to happen. And troponin is in the way. Troponin is blocking this active site. So all of this red is the actin. On top of our actin is something called troponin. It's actually 2 molecules. It's a troponin, tropomyosin complex. So I'll draw it with two little colors here. So it's literally blocking the active sites of where our myosin is going to bind. In other words, it's blocking the door, right? This is the dad that doesn't want his teenage daughter to go out. Nope, you can't come in. Or, I find sometimes my metaphors might offend people, and I don't mean it by any means. I really just mean it to be funny and to help you remember it. So I have a very risque way of thinking about this, and so it's not for the really serious person that might be offended. And I'm apologizing already if how I'm about to describe this makes you uncomfortable. It's not meant to make anyone feel uncomfortable. This is just the way my weird little brain thinks. And I find most students remember this the most because it's perverted, and we're all a little bit perverted, I guess. So I think of myosin as the male counterpart. You might think that's obvious by looking at my drawing here. I think of myosin as the male counterpart in this interaction. And actin is this demure little thing, right, that's crossing her legs, if you will. Right? So she's basically saying, no, you're not getting any. And all that needs to happen in order for myosin and actin to connect, right, is for this complex, this troponin tropomyosin complex, to get out of the way. In other words, for actin, who's sitting there like this, to say, hey, right? And the thing that does that, I mean, let's be honest, it's different for everyone. Some people like tequila. Some people like wine. Some people like beer, whatever, right? In this case, this actin molecule likes calcium. You give this girl calcium and she'll do anything, all right? So the calcium comes from the sarcoplasmic reticulum. Calcium comes in and binds to this troponin tropomyosin complex. And now she's loose. Now her legs are no longer crossed. And guess what? The myosin can get in there. Okay? It's all about connecting the two pieces so that this contraction can happen. Right? And so the active sites on our actin are protected by, I mean, you could think of it as like a chastity belt, right? Or she's just protected by the stress of the day, and she's just not ready for that. She has a headache, right? A little bit of calcium sprinkled in there takes all those inhibitions away. So tropomyosin moves when calcium is bound. So calcium binds to troponin. Troponin's sitting on top of the tropomyosin. And when calcium binds to the troponin, the whole thing moves. So calcium from the SR binds to troponin. You can do the same thing, troponin, the same thing with all of these words that we did at the beginning of lecture. Summarize. See if you can rewrite it in your own words, right? Calcium from the SR binds to troponin, which moves tropomyosin, which reveals the active site. Right, the active site on the actin. In other words, this myosin can't connect just anywhere on the actin. It has to connect at those active sites. Those active sites are being blocked. Here's what we got. Calcium being released, right? T-tubule charge is changing down here, that rapid depolarization. That depolarization opens up the calcium channels at the triad, right, at those terminal cisterna. Calcium's being released. Remember what's lying underneath of these structures are the myofibrils, right? Myofibrils, which are stacked end to end to end with sarcomeres, right? So you sprinkle a little bit of calcium on top of these sarcomeres, and what you're doing is removing the tropomyosin troponin complex. You're revealing the active site. So the myosin heads can say, okay, let's get it on, let's do it, right? And so that's what happens. That's this chain of events. Oops. There we go. So here is our active site being being blocked. The myosin head can't get in. Once we move, what calcium comes in, binds, it's going to remove that troponin tropomyosin complex. So, tropomyosin covers active sites, binds calcium ions, is small globular, or has 3 subunits.
Covers active.
Yeah, tropomyosin is what's actually in the active site of the actin. Troponin is where the calcium's going to bind. So this troponin tropomyosin complex kind of looks like this. And this is my actin. This is my thin filament. On top of it, here's my active site. On top of it, covering up that active site, is this. And on top of that is this. Our calcium binds there. This is our troponin. This is our tropomyosin. It's kind of stacked on stacked on stacked. So, troponin has 3 subunits. Which of the following does not bind to these subunits? Troponin. Look at where our troponin is. What's not going to bind to one of those subunits? Our troponin's on top, so we know that it binds to calcium. We know that it binds to the tropomyosin. It does not bind to myosin. That's the whole point, right? It's there blocking myosin from getting in. Myosin can't connect. So, choose this correct sequence of events. I'll give you a minute to read them and think about it. In preparation for contraction, what has to happen? Okay. Before any of this can happen, you have to have an action potential, right? That's the idea of excitation contraction coupling. You have to couple or pair the excitation, this stuff, with the contraction stuff here. None of this can happen if there's no action potential. So action potential arriving is the first one. There's only one of those choices that has action potential arriving at the triad first. Oops, the wrong way. Keep going the wrong way. There we go. All right, so the contraction phase begins with that actin site being exposed. The myosin head is cocked and ready. He's ready to go. He doesn't need tequila, right? And this ATP is actually bound to it. It's actually ready with ATP energy. In its high energy position, the head will actually cross over and connect with the active site. So it's already loaded with ATP. ATP's there. It's got potential energy. When it connects, the ATP is still there. That active site is revealed and it binds along, it's bringing along its ATP energy. What it needs that ATP energy for is the power stroke. Right now you see where I got these like analogies. I didn't make them up. Like maybe, Mr. Anatomy and Physiology was a little whatever. All right. So this power stroke is literally where these myosin heads, they just pivot a little bit. Imagine these little heads right here just pulling a little bit that way, right? But there's another piece of myosin here that's pulling a little bit that way also, and another one here that's pulling a little. And so if all of them are pulling, it can produce major movement, right? And that pulling, that little pivot, is what uses the ATP. Right? Myosin then has to bind to another ATP in order for it to detach. So let's kind of diagram this out. I'll use the board over here. So let's look at like the life cycle of a myosin molecule. It's loaded with ATP, right? We say that it's cocked, right? It's ready to attach. The active site is revealed. Myosin joins, or myosin connects. We say it forms a cross bridge, right? You can see why we call it a cross bridge. It's literally linking the thick and thin filaments together, right? Cross bridge. It still has that ATP. The power stroke, which is the contraction, uses the ATP. If you remember the hydrolysis of ATP, ATP forms ADP plus P, right? And ADP plus P can form ATP. It's this cyclical relationship, right? We went over this like the first lecture or something like that with ATP. So ATP is used up, meaning now we have ADP plus P, right? A new ATP has to bind. And it detaches. And then it's cocked and ready to go again. And then it binds. And then power stroke. And then a new ATP comes in and releases it. And then it binds, and then the power stroke, right? It's this cycle over and over again. The key here is that a new ATP has to be used. Otherwise, your muscle's gonna be stuck, right? Think about what happens after you die. You all know about this already because you've seen all the movies and everything. What happens to people's muscles after they die? There's a name for it.
Rigor mortis.
Rigor mortis, right? Why does rigor mortis set in? Think about what we just explained. Why does rigor mortis set in? There's no more ATP, right? Eventually your cells, which are dying, aren't making ATP anymore. So your myosin heads, which need a new ATP to detach, can't detach anymore. And so the body stays stiff. What eventually makes the body not stiff anymore? Anyone know? Yeah, decomposition. The muscle proteins are just decomposing, and so the actin and myosin are just falling apart, and so eventually everything, withers away. But that's why rigor mortis sets in, right? So, and roughly, rigor mortis sets in about an hour or two after death, right? So that means you have about an hour or two worth of ATP laying around without any other metabolic things happening in your body. Kind of cool. This is the same thing that we drew over there, but with the picture instead, right? So you have this concept of, you can start it anywhere, this concept of our myosin head is cocked and ready to go. It's loaded, locked and loaded. Okay, I'm ready to go. As soon as that active site becomes revealed, right? As soon as that troponin and tropomyosin get out of my way, I'm gonna get in there, right? It's revealed. The ATP is still there. ATP is used with the power stroke, right? A new ATP has to come in, reload it to detach it. So, the hydrolysis of ATP. Hydrolysis of ATP, what is that responsible for? I use the fancy word hydrolysis there.
Releasing the myosin.
So hydrolysis is digestion, it's breaking down. So in other words, when do we break down ATP? Right, in that power stroke, good. Recocking the myosin heads, yeah, okay. So I guess you could say up here, we're putting it together. You're breaking it down with the power stroke. So I guess the binding of ATP to myosin, this is responsible for what? When that ATP binds to myosin, what's happening?
Releasing.
Yeah, you're detaching, right? Releasing that myosin from the active site. release of ADP and P is occurring when?
Power stroke.
Yeah, that's the power stroke, right? Myosin heads return to their low energy or relaxed state when?
The release of the myosin heads from the actin.
Right, so they, at that power stroke, now they're relaxed, right? They're low energy, they don't have ATP, right? They're high energy, cocked and loaded with ATP. They're only relaxed right after that power stroke, and then they get reloaded again, detached, and are ready to go. The power stroke does what? Remember, the power stroke is the contraction.
Pulls the thin filaments towards the M line.
Right, that's what the contraction's all about, right? Pulling the Z lines together, pulling the thin filaments toward the M line, right? I bands, H zones, those are all shrinking in the process. So, the contraction cycle. I'm not going to cycle this out for you because sometimes I'm going to challenge you to do this on your own instead of just copying what I'm doing here in class. So I strongly suggest taking these words, these terms, these concepts, and creating your own cycle for it, right? This is what it is. The myosin head gets recocked, right? We already did that. Binds to an actin molecule, power stroke. It repeats. It binds to another actin, so on and so forth. So it's literally kind of walking down the actin molecule. This happens, so literally what it's doing, if let's say that our M line's over here, right? Let's say this is literally going to pull this. It's going to cock, power stroke, detach, cock, power stroke, detach. And with each time, this thin filament is going to move closer and closer to the M line. So in other words, we are used to thinking of it as, whew, this one big power stroke. But really, it's a million teeny tiny power strokes that produce this big action, right? These myosin heads literally walk down the line. It's like a conveyor belt, right? Pulling it through. Each time cocking and reloading, detaching, cocking and reloading and detaching to get that thin filament closer and closer to that M line. And that's literally, I mean, think about all the movements that you do, right? That's happening millions of times. It's crazy. So this is the cycle, right? This is the thing, pulling it, walking it closer and closer. You can see kind of what I mean by walking it. Notice here, it's on that very last actin ball on the second one. Now it's on the second one. Now it's going to go to the third one, and so on and so forth, just to kind of pull those thin filaments in. So think of like sailors like pulling an anchor rope, like pull, 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 right? Getting it tight, right? That's kind of how you can think of this, right? Now we have to imagine muscle relaxation and how relaxation occurs. So the motor neuron has to stop signaling in order for relaxation to occur. This is going to keep going until calcium is no longer available, right? Because when calcium is no longer available, troponin and tropomyosin hop back in place, right? She crosses her legs. She's like, no, I'm good, right? That's not going to happen until we don't have a depolarization up here. We're not going to have a depolarization up here until our acetylcholine's gone. Right? So this whole thing starts with our motor neuron and ends with our motor neuron. The motor neuron has to stop sending that signal in order for any of this to stop. Right? Calcium ions actually become pumped back into the terminal cisterna there. So calcium is released and all of this happens, but after it's done, calcium's taken back up. We mentioned acetylcholinesterase already, ACHE. This is going to recycle all of the ACH that's in our synapse. And the sarcolemma returns back to its resting level of polarization, right? The sodium-potassium pump is what does that. It puts everything back in place. It's going to pump all the sodium back out, pump potassium in, get everything back to negative 85. Right? And so that repolarization will take place all throughout the entire cell. Those calcium ions get pumped back into the sarcoplasmic reticulum, everything goes back to resting level. But it has to start from that motor neuron. The motor neuron has to stop sending that signal. If there's no calcium there, troponin and tropomyosin can actually block the active sites, and the muscle fibers can relax. But it's only if there's no calcium. If calcium's still there, it's going to bind and you're going to have a contraction. Right? So you have to take those things away. This is the whole thing, right? Muscle relaxation. It has to start up here. Start with that motor neuron. Put all the things back in place. Here's some multiple choice questions for us to think about. During muscle fiber relaxation, calcium channels in the SR close because we're turning off that motor neuron, right? We're getting our cell back to its resting membrane potential. The only reason those calcium ion channels opened to begin with is because there was an action potential, right? There was a depolarization causing those to open. During muscle fiber relaxation, where does calcium go? Yeah, it's pumped back into the SR. Acetylcholinesterase does what? So acetylcholinesterase recycles acetylcholine. What does acetylcholine do? Acetylcholine binds to the receptors and causes them to open, letting sodium go in. So if we don't have acetylcholine there, our ligand-gated sodium channels will close. During muscle fiber relaxation, oh, we just did this one. Just kidding. All right. We just sarcolemma, repolarization during relaxation. All right, that's what it's all about, getting it back to resting, getting it back to normal. Which aspect of muscle relaxation require ATP? What seems like it might use energy. Yeah, that's gonna use ATP, right? Anytime you have to pump something, right? Pumps always use ATP, right? The sodium potassium pump uses ATP. Cool. We talked about rigor mortis already. 3 to 4 hours after death. I said 1 to 2. It's somewhere in that range, right? It's basically the pumps that drive the calcium are no longer working, so there's ATP just kind of lying around. Once that ATP is used up, there's nothing to detach them. There's no ATP to do this, so they stay attached. The muscle fibers are unable to relax. Myosin can't detach without ATP. So everything kind of stays tight until 48 to 72 hours after death, and that's when the muscle protein fibers just begin to eat away. Which, if you think about our like ceremonies and what we do, like, you know, when the viewings are, when the funerals are, they're still kind of during this period of rigor mortis, which, you know, if you've ever touched someone at a viewing or at a funeral, usually touch them at a viewing, not at the funeral. Anyways, so this is the big picture of relaxation, right? It's turning everything off, working backwards. We have to reattach our troponin and tropomyosin. We have to take away the calcium. We have to repolarize everything. We have to take the acetylcholine away, etc. It's working backwards. That's what relaxation does. So how do our muscle cells get all of that ATP? Because sometimes ATP is not available. So there's a couple of ways our muscle cells are going to get ATP. ATP is the major energy molecule for all of these contractions, right? It's powering the sodium potassium pumps. So all of the resting membrane potential, all of that remaining there, right? Pumping the calcium back in, it's all done through ATP, right? Releasing the myosin heads, all ATP. We need ATP for all of these things. We can create ATP through a couple of activities, right? Through systolic reactions, catabolic reactions, Oxidative reactions in the mitochondria. We'll look at all of these reactions in detail in AMP2. These can all occur all at the same time in our muscle fibers. So in other words, it's kind of like a short order cook. Your muscle fibers are saying, hey, we're using heck a lot of ATP over here, contracting, so can I order some more, right? And your muscle cells have the ability to create more ATP because of the nutrients that are stored in them, right? So let me just go back for a second. Oh, I should have tried to go back. So one of the things, glycolytic catabolism, look at that word, glyco. What's that making you think of? Sugar, right? Lytic, like breaking down, catabolism or breaking reactions. Glycogen is our body's stored form of glucose, right? Our body eats too much sugar, we first store it as glycogen. And if those stores are filled and we're eating too much, then of course it's converted all into triglycerides. But glycolytic catabolism is taking the glycogen that's stored in your skeletal muscles and breaking it down and using it to make ATP. So like I said, it's like that short order cook. Hey, I need to order a cheesesteak and fries. That sounds really good right now. Hey, I need to order some ATP from glycogen, right? Breaking it down. So creatine phosphate is another way your cells can get ATP. So if for some reason immediate ATP energy is not possible, maybe you ran out of glycogen, maybe you just don't have enough oxygen available, creatine phosphate is a backup plan. So creatine phosphate is stored in the cytosol of your skeletal muscle fibers, so it's there waiting to be used if needed. You may have heard of creatine if you've ever shot for like protein powders or gone to GNC. It's the same idea, right? Giving your body this extra quote-unquote energy. Now, I'm not going to say those supplements work or whatever because it's all in how your body absorbs and uses the nutrients, right? Just because you're putting it into your body doesn't mean you're not just peeing it right out, right? So there's a couple of chemical reactions that have to take place in order for you to even use the things you're putting in your body, like those supplements. But the creatine phosphate that's already in your cells can be used to make ATP. Now remember, ATP is, I'm going to erase this and I'm going to take a look at our ATP hydrolysis a little bigger here. This is how our body uses ATP. So normally ATP is broken down into ADP plus P, right? And so this is how we use energy, and this is how we store energy, right? Well, creatine phosphate can do the same thing because creatine phosphate is basically taking the P from creatine and giving it to ADP. So it's literally adenosine diphosphate is APP, adenosine triphosphate is APPP. So you're literally taking the P, taking the phosphate from creatine, and donating it to ADP to create ATP. So this can happen pretty quickly. And so your body, again, will choose to do this over other means of getting energy. But it's not like it's going to sustain you for very long. It's just a short amount of creatine phosphate that we have. So this is looking at that equation, right? ATP energy is usually derived from ADP plus P. But sometimes we can get that third phosphate, that third P, from creatine phosphate to get the ATP that we need. We can also do glycolysis. So glycolysis is a series of reactions. It's anaerobic. It does not require any oxygen. And so what it's doing is it's breaking down glucose to get pyruvic acid or pyruvate. So